Hello, my name is James Kai. I'm from Roche Innovation Center, New York. I'm also here representing two other authors, Louise and Xiaoxi. The title of our presentation is Rapid Mining of Scientific Literature to Enhance Drug Repurposing Process for COVID-19. So what's the problem that we are trying to solve? When we face the current COVID-19 pandemic, there is an urgent need to identify drugs for treatment. And our best bet is to find the existing drugs that can be repurposed, because it would have taken too long to develop new drugs. One approach is to identify drugs that target human proteins that play key roles in the virus life cycle, instead of targeting the viral proteins. This has higher likelihood of identifying existing drugs. The most direct source of such information is in the structured knowledge bases, such as DrugBank, TTD, PharmGKB. However, because they rely on human manual curation while being very accurate, they can be limited in both the coverage and also to stay current. So we asked the question, can we leverage the latest findings of what human proteins interact with the virus and therefore identify drugs that inhibit these proteins? We started from three lists of human proteins. The first is the list of proteins that are known to be involved in the entry of the virus into the cell. And the second is the list of proteins tagged as COVID-19 related in PubChem. And the third is a list of proteins discovered in a cell-based study in the lab. Our goal here is to find existing drugs that can target these proteins. We use an approach called Snowball relation extraction. This is a semi-supervised bootstrapping NLP approach to extract relations between drugs and genes or proteins. The advantage of this approach is that it only requires a small number of training examples to initiate the process, allowing more training examples to be added in subsequent iterations. This slide explains how snowball relation extraction works. The whole process has two phases, the learning phase and the extraction phase. In the learning phase, a small number of example sentences and the corresponding tuples of the chemical inhibit gene relations were parsed for dependency grammar-based feature generation, which I will explain in the next slide. This produces a matrix of one-hot vectors of the features, which I will also explain in more details in the next slide. In the extraction phase, a list of protein names is given to a public tool called PubTator, which searches PubMed and generates sentences that are annotated with gene and chemical mentions. Again, these sentences are parsed to generate a series of one-hot vectors, one for each sentence. The vectors are then compared with the seed matrix produced in the learning phase for close matches using the k-nearest neighbor identifier. When there's a close match, the new sentence was then recorded together with the tuples of gene, inhibit, and chemical. Vectors that have close enough similarity to the seed matrix were also added to the matrix to expand the relation patterns to extract even more instances of the new pattern in the next round. Here I want to explain how grammar-based feature generation works. So we start from an example sentence that contains the relation between a gene and a chemical, represented in the tuple of chemical inhibit gene. Next, we parse the sentence with the PubTator to identify the entities from the sentence, and then we perform the dependency grammar parsing using a tool called NLP4J to generate features, which are then captured in a one-hot vector. So each element of the vector is a feature that describes the dependency connection between two words, and each vector describes a pattern of a linguistic relation between the gene and the chemical. So what did we find? We found that starting from the list of proteins that are involved in the entry of the virus into the human cells, we were able to identify 23 drugs. And it's very good to know that 9 of them are already in clinical trials for COVID-19. Using this approach, we also identified 5 ACE2 inhibitors and 3 serine protease inhibitors that are not recorded in the knowledge bases. When we looked at the proteins tagged as COVID-19 related in the PubChem database, we discovered an additional 40 substances that are not covered in PubChem. Thirdly, when we looked at the list of proteins published in this Nature paper, we not only confirmed some of their findings, but also found 53 new drugs that target the proteins that were discovered in this paper.